Hello and welcome back for one more video in Chapter 7 in which we are looking at the theory and practical usages behind spectrophotometry and concentration assays. So in the previous video we really got into discussing some of the general basics, the theory behind how spectrophotometry works, and some practical applications. In this video, we want to focus specifically on using the spectrophotometer to measure both protein and DNA concentration. So, I said it before, but at this point in the semester, we have already done a Bradford assay, which is using the spectrophotometer to measure the concentration of total protein in solution. Well, part of the issue with that is that proteins by themselves are not visible, right? So proteins in solution should be colorless. So when we do the Bradford assay, we add a dye to it that actually binds to the proteins and causes them to turn blue. So the problem with doing the Bradford like that is that just on its own, you can't determine the total protein concentration. You kind of have to have a standard to go off of. So that is why for the Bradford assay, and you can do this for a variety of other spectrophotometric assays as well, we used what is called a standard curve. And to do that, we used a solution of protein that has a known concentration. In that case, we used a 1 milligram per milliliter solution of bovine serum albumin, or BSA. So what we did with that is that we made a variety of dilutions of BSA. We had one tube that didn't have any BSA in it. We had another tube that just had 1 microliter of BSA in it, another one that had 2, another one that had 5, and another one that had 10. So starting with no BSA, we had 5 tubes that became progressively more and more concentrated with total protein. So along with our unknown samples that we're trying to figure out the concentration for, we also had these standards that we did the Bradford assay on alongside with them. So the advantage of this is that because we know the original concentration of the BSA, we know the concentration of the BSA dilutions. And if we can get absorbance values for those dilutions for our standard curve after we do the Bradford assay, we can really compare the absorbances of the standards and our unknown samples, and we can figure out the concentrations based on the absorbances of our unknown samples. So if you look at it graphically, your standard curve should fall on a straight line like this. So your 0 tube of BSA should be right here. It should have no absorbance because this is usually what you blank the spectrophotometer with. Your lowest dilution should be about here, then this, then this, then this, then this. So you can make as many dilutions as you want, but the relationship between the concentration of the standards and the absorbance of the standards should be linear, as we said before. So basically, we can, using something like Microsoft Excel, we can plot out the known concentrations on the x-axis and the measured absorbances from the Bradford assay on the y-axis, and we can plot the line. And the advantage of this is that if we know the absorbance of our unknown samples, we can just figure out where that matches up on the line and figure out what the concentration is. So that's really to our advantage here. So you can do it that way, or if you want to be a little bit more mathematical about it, programs like Microsoft Excel will perform linear regressions for you in which it gives you the actual algebraic equation of the line in the form of y equals mx plus b. So you would just use the slope of the line to figure out the concentration if you know the absorbance. So the Bradford assay is absolutely preferred if all you've got at your disposal is a visible spectrophotometer, because the lambda max for the Bradford reagent when it is bound to protein, 595 nanometers, that is squarely in the visible range. So Bradford is what you'll want if you've got a visible spectrophotometer. But like I said, if you're not going to do the Bradford assay, you can't measure protein concentration if all you have is a visible spectrophotometer. But if you've got a UV spectrophotometer, you don't necessarily have to do the Bradford assay. You can if you want to, but you have another option at your disposal. and that is even a colorless protein solution will absorb UV radiation, specifically at 280 nanometers. And this is all due to two particular amino acid side chains, those of tryptophan and tyrosine, which are pictured here. So if you look at the side chains for each of these, you'll notice they each have an aromatic ring, which is this kind of hexagon-looking shape that has alternating double bonds. So because of the resonance nature of these ring structures, they actually are capable of absorbing UV radiation, meaning that if we are measuring spectrophotometrically at the appropriate lambda max, which is 280 nanometers, we can apply all the same spectrophotometry theory to determine the total concentration of protein in solution. But as it mentions here, you would obviously need to take care that there is nothing else in your sample that absorbs at 280 nanometers, something like NAD+. So if you do have a reason to suspect that there may be contaminants that would throw off the accuracy of your measurement, then maybe it would be better to just do the Bradford assay. So you have to be a little bit careful when you are using UV spectrophotometry to measure the concentration of proteins. And that is because you cannot really use the Beer-Lambert law to determine the exact protein concentration at 280 nanometers. And the reason for that is because different proteins have different numbers of tryptophan and tyrosine residues. So as long as two different proteins have different numbers of tryptophans and tyrosines, those two proteins are going to have different extinction coefficients. So you can't really use the Beer-Lambert law if you don't know exactly what the extinction coefficient is. So yet another reason why it may be preferable to just do the Bradford assay. But the 280 nanometer UV method of measuring protein concentration should be a good and quick and easy way to get a general idea of how much protein is in solution, although we certainly have laid out several reasons why you may just want to do the Bradford assay. So it turns out that you can actually use UV spectrophotometry to determine the total concentration of DNA in a sample. So you're going to use a slightly different lambda max value here, which is going to be 260 nanometers, which is also in the UV range. So the nice thing here is that you actually do not need a full-blown standard curve necessarily, but having at least one known standard should be helpful. And your textbook takes you through a couple of examples in which you use cross-multiplication using the absorbance of your standard against the absorbance of your unknown sample, and you can just use that cross-multiplication to very easily figure out the concentration of your unknown. If you assume that, say, something like 50 micrograms per milliliter DNA absorbs at a absorbance of 1, then you can use that cross-multiplication to pretty easily figure out the concentration of your unknown if you know the absorbance that matches it. So the last thing that we want to mention here is something that should be very useful if you get to the point where you're doing a lot of DNA purification. So this is a measure of the purity of DNA in a preparation of DNA, and this is what is called the 260 to 280 nanometer ratio. So we've already established in the previous two slides that DNA absorbs at 260 nanometers and proteins absorb at 280 nanometers, both in the UV range. So if you're performing a type of DNA extraction in which you are getting DNA purified from cells, you're going to have a little bit of contamination. So it's not going to be just 100% DNA. There may be a little bit of RNA, there may be a little bit of this and that, but your most common contaminant is going to be proteins. There are a lot of proteins that really like to stick to the DNA. So since we know that DNA absorbs at 260 and proteins absorb at 280, if we measure both of these absorbances, both at 260 and 280, we can use the ratio of those two absorbances as a relative measure of the purity of the DNA sample. So what we want here is we want to get both absorbances, both the absorbance at 260 and the absorbance at 280, and then just have the 260 in the numerator, which is roughly speaking about the concentration of the DNA, and the absorbance of 280 in the denominator, which is roughly related to the total concentration of protein. So whatever ratio we get should give us a general idea of how much protein contamination there is. So the idea here is that the more DNA and the less protein there is, the higher the ratio should be, because the more DNA, the higher the numerator is, and the less protein there is, the lower the denominator, both of which would cause the ratio to be a bigger number. So practically speaking, you generally want the 260 to 280 ratio to be at least 1.6. If it's any less than that, then you've got really undesirable levels of protein contamination. If you really want to use purified DNA for specific applications like transformation and transfection, then you usually want a little bit better than 1.6, maybe something in the 1.8 to 1.9 range. In that case, of that range, you're both minimizing protein contamination and you're minimizing RNA contamination. As your book mentions, having a 260 to 280 ratio that is greater than 2 indicates that you've probably got a lot of RNA contamination. All right, so that is going to do it for Chapter 7, in which we hopefully learned an awful lot about not only the theory behind spectrophotometry, but also a lot of great practical uses for it. So spectrophotometry is going to be a very, very important and very fundamental skill that you're going to use pretty often when you're in a research laboratory. So it's definitely good to get lots of practice with it and to make sure that you understand the theory behind it. So that is going to do it for me for now. I will see you again in next chapter of Chapter 8, in which we will start talking about some applications of recombinant DNA technology. So I will see you next time.